we are ready, yes?
Yes.
Okay. Now we are starting.
Okay.
Okay, now, first of all, by the way, which university you are in?
Oh, Stony Brook University.
Okay, in which state it is there?
It's in New York State.
Really? New York
Yeah.
City, okay. But the what about the rank of the university, Fatin?
Uh, there like
You
rank.
just got admission into US, so you didn't go for the rank and these
Oh
things,
no,
yes?
no. The thing was, what do you call that? When I looked at this, wait, but I looked at biomedical engineering, and I think it was medical engineering. I looked at biomedical engineering, and it came in as 64.
64 is not bad at all, you know.
But yeah, it's oh, it's six. Sorry, it's 64 in general throughout the U.S. But for only for
Okay.
engineering, it's 25. But I didn't go in for engineering, so I went in for I went in for biochemistry.
Okay, no problem. Then what was the name?
Stony Brook University. It's not
Stony Brook
difficult.
University.
Yeah, it's not difficult to get in.
Yeah, okay, okay. So anyway, we are starting with this chapter that is gene expression. Yes.
Yes.
So we will start like learning objectives. Obviously, I'm not going to do with you, correct?
Yeah.
Okay.
Yes.
Now this we will be just moving. I'm just will be moving down to the main topic. Yes.
Yes.
Okay. Okay. Now, first of all, I mean, what is the process of protein synthesis? So this is a simple thing. Like, what is protein synthesis? The DNA is transformed in the form of RNA,
RNA.
which is
Yeah.
messenger RNA, yes, and
Yeah.
then messenger RNA is transformed into protein.
Mhm. Yeah.
Correct.
Yes.
So this
I'm
once
not
the
saying much. Sorry, sir. I'm not saying much because it's recording. So I'm like,
no, you keep on. No, you
okay,
keep your inputs in. That doesn't matter.
okay,
Because after that, what you will do, you will be sending this recording to me in my email. So this is again a news for you. Like I have started my website.
okay,
Okay, so what I'll do, I will be uploading this video, parts of this video into my website. Okay, I
okay,
will upload it there. Okay,
okay.
so don't worry if you interact because most of the teachers who upload their presentation,
Hmm.
they just talk on their end, yes, but there is no
Yeah.
mutual participation, that likes live session. So I want that it should be a live session, yes. So
Okay.
you keep asking me the question, don't worry.
All right, sir.
Okay, now the thing is, once we are talking about the protein synthesis, once this the DNA information is transcribed or translated in the form of protein, this is known as gene expression. What is gene expression? When the information from gene is
Trans
translated into
into
or converted
protein.
into proteins.
Yes.
That's all correct. Now,
Yeah.
transcription and translation already we learned in A
Yeah.
levels, even O
Yeah.
levels also, yes. Like
Hmm.
transcription is the conversion of DNA into messenger RNA, and translation is converting this messenger RNA into protein. Isn't it
Proteins.
polypeptide?
Yeah. Polypeptide.
No issue, correct?
Yeah. This is
Okay.
like we've done it twice now. I've done it twice now. So
Okay. Okay, no problem. Okay, now the thing is, what is the name and what is the nature of the gene and how do genes control phenotype? Now, actually, this is interesting kind of thing. Okay,
okay.
so because it was the initial phases, yes, initial phases.
Yes.
So there is one concept which they are saying like one gene, one hypothesis, one gene, one polypeptide, and one gene, one amino acid.
Hmm.
So
Yeah.
each gene is responsible for one particular
Amino acid.
just amino acid or polypeptide. Okay, so now the study metabolic defects provide evidence that genes specify protein. They are specific, correct? So each
yeah.
gene is specific to make one particular kind of protein. Now, what actually they have done, I will show you this thing. The tools I'm going to use now, okay? For example, they are saying like the protein, that phenylalanine. It has to go through different kind of channels, and ultimately it has to end up with a harmless substance, which is water and carbon dioxide. Correct?
Yes.
All these processes will take place in which part of our body?
Ribosomes, right?
Liver. No. Liver.
Liver. Oh, sorry.
Breakdown
Breakdown
process,
of
it will
protein.
take place, breaking down, everything will take place inside the liver. But the thing is, once we are talking about phenylalanine, it will turn into tyrosine. Tyrosine will go into hydroxyphenol pyruvic acid, then HA or homogentic acid. Yes, they are written HA. Yes, and then it converts into a new what is actually melyl acetoacetic acid, okay? A derivative between HA and carbon dioxide and water. So actually what is there? HA is what? It is harmful. It is poisonous, toxic. So we have to convert into harmless substance, which is actually the function of normally the organ which is involved, that is liver. But obviously this conversion need some kind of enzymes. So if this enzyme is not present, which is homogentic acid oxidase, then what happens? There is one condition which is, this condition is acaptonuria. Acaptonuria or captonuria. Yes, this is a condition when a person is not producing this as this enzyme. Yes, so this HA cannot convert into water and carbon dioxide. So once a person urinates, once he urinates or she urinates, it turns into dark color, black color. It is very horrible kind of condition. And why this is happening? Because some of the changes has taken place in one particular gene, the gene which is responsible to produce HA, and because of this thing, they are saying like they are alkaptonuria, loss of an enzyme in phenylalanine, like phenylalanine has to get converted into what in the process, okay? Tyrosine metabolism pathway causing accumulation of HA, homogentic acid, okay? Which accumulates in joints, causing pain and found in urine. So once a person who is suffering from this, that person get what? Darker urine or black color urine. It happens in children, okay? Childhood, it is more prominent. Okay. So now actually they wanted to see how one gene is controlling all this. So they wanted to, what has happened? So they found there is a problem in one gene, the gene which is not producing this enzyme, okay? This enzyme is not being produced by the gene. So that is why this conversion of HA into carbon dioxide and water is not taking place. So this condition happens. Okay. They have to just verify. Okay. Now next slide we move. Okay. Okay. All right. Yeah. You just got that because my purpose is not just to read. At least something you get it. You have your notebook with you? Yes, sir. I'm typing everything down in Word. So. Okay, good. Now the point is here we should be knowing the next topic. If we go next slide, in fact we are going. In fact, just 1 minute. So I'm removing all this and I'm moving to the next slide now. Okay. Okay. Now, if we move to the next slide. Okay. Now, if you go with this slide, they will be showing now one gene, one enzyme experiment. Beadle and Totem, they are very famous scientists, okay? Like one gene, one protein, or one gene, one polypeptide, or one gene, one enzyme. This experiment they wanted to explore. So they conducted the experiment, okay? What they took, that neurospora cells. They took neurospora cells. Okay? Neurospora, here, the neurospora, I will be showing you. Neurospora here.
Yeah, it's a fungus,
I will
right?
be annotating. Yeah, I will be just annotating it so you will understand. So this part, neurospora is there.
Mm-hmm. Yes. Sir.
Okay.
Yeah.
So first, what are they doing? They are using the neurospora cells. They were placed in a complete medium. What does it mean by the complete medium?
Complete medium.
A medium which contain all the required nutrients. Required nutrients for growth.
Okay.
A
I'm
medium
just going
which contain all necessary nutrients for the growth.
Okay.
Nitrogen, phosphorus,
Nitrogen.
sulfur, or anything,
Amino.
whatever is required for the growth, everything is there in that medium. So what they place all the neurospora cells into that complete medium, so they grow healthy.
Okay.
After that, they expose these spores or neurospora cells to X-ray radiations. So what will be causing radiation will cause some kind of mutation, obviously. Radiation will cause mutations. So different neurospora cells, they will be having different kind of mutations, isn't it?
So the X-ray radiation will cause like different types of mutations because it doesn't affect the same place.
Yeah, right? they will cause, okay, just remember they will be causing the mutation.
Okay.
Okay, then what they did, look, this neurospora cell is there. Okay.
Yes.
If they place this neurospora cell in a minimal medium.
Okay.
What is a minimal medium? A medium which have limited supply of nutrients. Limited supply of nutrients. Yes. Okay, basic supply of the nutrient.
Yeah. Just
okay? Not complete range. So what happened? There was no growth.
Okay.
Then what they did, the same neurospora cells which were exposed to the X-ray radiation, they divided them into 3 groups, 3 categories.
Okay.
Okay, what is the category? In one minimal medium, they provided the minimal medium, but they provided along with them valine, one amino acid.
Okay.
So what does it mean? It means that amino acid did not put any kind of effect on the growth. There is no growth. Then second minimal medium they used and they added lysine, another amino acid. Nothing happened, no growth.
So they added lysine.
Got it or no?
Yeah, they added valine, there was no growth. They added lysine, there was no growth.
There was no growth. They added lysine, there was no growth. Okay. And then what happened? They added along with the minimal medium arginine.
Arginine. Okay.
And they found there is a growth.
Okay.
What does it mean? What does it mean? Due to the radiation, the gene which is responsible to produce arginine was
not present.
Was missing or it got mutated.
Okay.
So arginine was not produced. So if you provide arginine, so this neurospora cell will start to grow. Okay. So it means what a Beetle and Totem model or experiment. What was the experiment? The basic of that experiment was one gene, one enzyme, or one gene, one amino acid, or one gene, one polypeptide. So what they say, one particular gene is responsible to make one particular kind of polypeptide enzyme. So it means a gene which is responsible to produce arginine, it was damaged. So that is why if you replace this with arginine, then the growth will take place.
Only if you replace it with arginine, the growth will take place.
Yes.
Okay.
And then what happened? They now look, in the minimal medium, they did not put arginine now, but they put the wild type cell. Which wild type cell? Neurospora, without exposure to the radiation. Okay. Since their arginine producing gene was not destroyed, so without adding arginine, the growth has taken place.
Okay. So because in those cells, the arginine could be produced. It yes. Was okay. So
because like the gene which was responsible to produce arginine, it was not damaged. Okay. Since it is not damaged, so this is not required. So it's mean there is here what this experiment is showing. This experiment is indicating the arginine producing gene is damaged. So you have to add arginine, only growth will take place.
Okay.
Here, because arginine producing gene is not being exposed, there is because this neurospora, this is the wild, this one we have taken directly here.
Yeah. So
okay,
that's like
before, so they are original. So if you take the original neurospora cells and give them the wild, the minimal medium, growth medium, they will start to grow.
okay.
You understand?
Yeah, this is
So it's
this
mean,
is okay.
yeah. No, go ahead.
So it can be called the, oh, that is the control, right? Where
That is the control, because you have to justify, yes, this is happening because, look here, if you just provide the minimal medium on both sides, will the growth take place here? Only minimal medium?
no.
No. Here you provided, look here you provided the minimal medium. In this case, you provided the minimal medium here.
Yes.
You provided the minimal medium, there was no growth.
No growth. But when they added the
once you arginine, then you are exploring different kind of amino acids. So you tried with valine, you tried with lysine, you can try with alanine, you can try with glycine. So if for example, I'm just giving you an example. If you here minimal medium plus arginine, there was no growth. What does it mean? Arginine producing enzyme gene is okay. It did not get changed. But if I put here glycine, I'm just giving you an example. Glycine. If I put glycine here and growth takes place, what does it mean? The glycine producing gene is damaged.
Okay. If it grows in
here, what will happen? So in this way, what is the basic outcome of beetle totem experiment? The basic outcome is one gene is responsible for one specific protein or enzyme or amino acid.
One gene, one
enzyme. One polypeptide hypothesis.
Okay, one gene, one polypeptide hypothesis.
Yeah, so here if we see it here, so there were one-to-one relationship between genes and specific enzyme. It should be possible to create mutants that are unable to carry out specific enzymatic reaction. So this mutant, it is unable to carry out production of arginine. Yes, this is the mutant now.
Yeah, it's a mutant.
So you can create a mutant. Maybe you will create, maybe this mutant, any other mutant you take from here, maybe this mutant is for glycine. This mutant is for, maybe this is for valine. So it depends. So once you are exposing all these neurospora cells to X-ray, so different cells, they will be mutated differently.
Okay.
You understood?
Yeah. This part is fine now. Okay.
Okay. Now I'm moving next now. This next slide.
Next slide.
Okay. Next is this one.
Genetic code.
Genetic code. Now this genetic code, once we are talking, here in your university, they are using the term redundant and not ambiguous.
Yeah.
There is one, the term that we use in GCE, okay? What are those terms? That is non-overlapping and?
Overlapping.
No. One is the characteristic feature of the genetic code is it is non-overlapping and degenerate. These were the two terms that we learned in A levels.
2 years, yes.
You recall?
Yeah, it's 2 years ago, but yeah.
Yeah, but anyway, you can recall it. I'm recalling after so many years. So this is just 2 years, man. Come on, you are younger.
Yeah, no.
So redundant is actually alternative. Like if we see redundant, and so this is, what is the meaning of degenerate? Degenerate means one genetic code. There, one amino acid can be produced by more than one genetic code. You remember this thing, yes?
Yeah.
Look here, like UCU, UCC, UCA, UCG. Yes, it is producing serine. So these 4 genetic codes, they code for one amino acid.
Okay.
So this ability is known as degenerate.
Okay. Okay.
Or
Yes.
you understood? Yes.
Yes.
So here redundant because most of the amino acids are specified by more than one codon. So redundant and
Redundant
degenerate
and
is the same
degenerate.
term.
Redundant and degenerate, then ambiguous and non-overlapping.
Yeah,
Not ambiguous.
but not ambiguous. Not ambiguous.
And
So not ambiguous means non-overlapping.
Non-overlapping. Okay.
Okay. So
Yes.
now non-overlapping, do you remember or I should explain it to you?
1 second. I just need to recall. It means that like.
No problem. You know, wait, I will do it for you. Okay, because we are saving time. A,
Okay.
C, C, G, C, C, A, G, A, G, U, for example. This is there. We will
It's
always
like
read how many.
The codons will not overlap, right?
Yes, non-overlapping. So it will be A, C, G. And then next will be what? G, C, C. And then will be A, G, U.
Yeah, triplet codon.
Yes. So it cannot be read like this. A, C, C. Then we cannot see it. C, C, G. You cannot do it.
Okay.
Or C, G, C. You cannot do it. After every 3 codon, new codon will start.
Okay.
So this is non-overlapping. Not ambiguous.
Non-overlapping and not ambiguous. I'm just typing. Sorry, just typing everything.
No
so it's
problem. You
taking
take
a
your
bit more
time.
time.
No problem. Take your time. And one more thing is there. One more thing. So you know the start codon, yes?
Yes.
AUG is always the start codon. We learned this thing, okay?
Okay.
AUG is always the start codon, and there are 3 stop codons, yes?
Yes.
There are 3 stop codons, and these 3 stop codons are this one, UAA, UAG, and UGA, yes?
UAA,
So what are stop codons? Stop codons are also known as nonsense codons. Why we call them the stop codon? Because they don't code for any amino acid.
Yeah, they just tell the amino acid to stop.
And start codon.
Okay.
Start codon always come in the beginning of a messenger RNA, and every messenger RNA starts with AUG, methionine.
Methionine. Yes.
MET, yes? AUG codes for methionine. MET, methionine. Here, it is here, like you can see it. This part.
Yeah, MET. And then there's renin, arginine, lysine. Oh, wait. I think if I can find the A-level textbook, then I think I'll be able to.
No need now, because we are saving our time. Your textbook is, I am your textbook now.
Okay.
You know, so next time once we have to go for, so you can just find out your A-level textbook and you can just revise.
Okay.
Okay. But actually, if you don't revise also, this is okay, because we are revising exactly now, isn't it?
Yeah. Yes.
So are you clear about this?
Yeah, clear.
Redundant and not ambiguous. Redundant mean degenerate. Redundant mean one amino acid can be coded by more than one genetic code. So what is the advantage of this thing, Fatim?
Advantages. If there are like slight mutations because of crossing over, it won't, affect the change. If it won't affect the body as much.
Very good. Yes, that is what. If by any means, by any chance, mutation has taken place, So this mutation may be the mutation which has taken place. It has not caused any kind of harm, correct? Because it coded for the same gene. So, so this is the benefit. Yes.
Yeah. Okay.
Okay. Now I'm moving to the next. You are clear about these two terms, yes?
Yes, sir.
Okay. Now the next slide is, yeah, obviously the genetic code is universal for all, starting from bacteria up to the complex animals. So
Yeah,
what
it's the
is,
same.
why is this significant?
Because then we can, I don't know how to explain.
Then
So
look
does
here,
this
you
mean
can
that
use, look here, in genetic engineering,
everything?
you can use the genes of one organism and you can insert that gene into any other organism.
Yeah. So does this mean that there was like they
In
had some kind
evolution,
of relationship at the start? You know,
yes,
they
that
all.
is what you can say like this. All the word, once we are talking about the vertebrates, so how, what is the basic, you know, how we know that all the vertebrates, they are very close together genetically. So pentadactyl limb, you remember?
Yeah, pentadactyl limbs and then the spinal cord.
Yeah, spinal, presence of spinal cord
Yeah.
or backbone, yes. But
Yes.
pentadactyl limb, to our level, we have to talk about pentadactyl limb. Spinal
Pentadactyl
cord,
limb.
the only kids can talk about, yes.
Yeah. Yes. Yes.
Yeah, so it is significant. Why? Because it has universal application. So if we get, for example, if we get a genetic code from a plant and that genetic code is, that gene is coding for one amino acid, which is valine. If the
Yeah.
same genetic code is present for human and same code is there, so it will be coding for valine. So it becomes very easy for us to identify the protein structure, primary structure of the protein.
Okay.
You
Yeah.
understand? What is
Yeah.
the primary structure of the protein? It tells us, gives us information how many amino acids are there and what is their sequence, isn't it? So it
Yeah.
becomes
Number of amino acids
significant
and
in that
the
context,
whole
correct?
sequence. Yes.
I'm moving next now.
Okay.
Okay, now the next slide is, here, this slide is about gene expression. Just 1 minute, gene expression and transcription, correct? Now,
Yes.
only one thing which is additional here, only one thing which is additional here, which I wanted to teach you, but because it was not there in our portion, so I didn't teach, okay? This is
Okay.
a you know, a primer region is there and a promoter is there.
Yeah. Primary and promoter regions.
Primer and promoter, okay, I will tell you now, just 1 minute, okay? There is a promoter kind of thing, okay?
Okay.
Here, this is the promoter which they have mentioned here, okay, promoter. So
Okay.
what is promoter? You know, RNA polymerase is there.
Yeah. Yeah. RNA polymerase.
How the RNA polymerase will understand that I have to start the process of transcription from here. So if the promoter is there, RNA polymerase, it is acting as a signal for RNA polymerase, attach here and start the process of transcription from here.
Okay.
This is very important part, okay?
All right.
You understood, yes?
Yes.
And the direction of this formation of messenger RNA is 5 to 3 direction. You know this 5 to 3 direction, yes?
Yes.
What is 5 to 3 direction?
Sorry. What do you call that? It starts at the fifth.
What fifth?
The fifth code. The fifth. I don't know the
Look, fifth carbon, yes. So
Yes.
this is pentose sugar, yes.
Pentose sugar. It starts at the fifth carbon in the first one.
Yes,
And
this is now actually this will be this is number 1, yes. Number 2, then it will be number 3, then it will be number 4, and fifth carbon will be here, yes?
Yes.
Somewhere here. It
Yes.
is fifth carbon. This is fifth carbon here, yes?
Mhm.
So at number 5, what is there? Here is what? Here is nitrogenous base.
Okay.
Here is what? Phosphate group, phosphate.
Yeah.
Fifth number. And here is what? OH group, hydroxyl group.
Okay, so
Please
they
make note
start.
of this, okay? This is because before you didn't need this one. Maybe now you require this part.
Yeah.
So this is a nucleotide, yes?
Yes.
Ribose sugar, nitrogenous base, and phosphate group. So phosphate group is present at number 5. So number 3 is hydroxyl OH group, correct? So always addition of new nucleotide will take place from this end, from this end. So this is 5. So chain will, new amino acid will grow on this side. So that is why it is 5 to 3 direction. We always write like this.
Yeah. So it starts at, yeah, it starts at the fifth with the
fifth end is always empty. Nothing happened. No, nothing can add there at the fifth end.
Yeah. Okay. And then same thing with the third end. That's when it ends.
So it keep on adding. So third end it can increase in length from here also and here also.
Okay.
Correct? They are complementary. 5 to 3. One DNA strand is 5 to 3 this way, and the other DNA strand. You know this complementary nitrogenous base pairing. Yes.
Yeah.
Okay. Now, what is the role of the promoter? It actually guides RNA polymerase to start the process of transcription from here. It will attach there and start to go for the transcriptional process. Okay, now once we are talking about gene expression, during the transcription, messenger RNA has to be formed, isn't it? Messenger RNA has to be formed. Once this messenger RNA, just 1 minute. Once this messenger RNA will be formed, yes?
Okay, formation from RNA.
So here.
Yes.
Transcription is a template-directed polymerization, similar to DNA replication. What is template-directed polymerization? Because one strand acts as a template strand and other strand is acting as coding strand. Remember?
Yes, yes. The first
2 strands of DNA are there.
Yeah, one is the
coding strand.
The other one
is template strand.
Yes.
So one acts as a template, and the coding strand is responsible to make its own copy, its image, mirror image.
Yes. Okay.
I will be, and this, transcription process is actually divided into 3 main stages. Okay, we call them the first is actually initiation. During initiation, what is involved? Promoter and RNA polymerase. So what will be the start point?
Start point will be the promoter.
Start point will be AUG. Yes, AUG will be messenger RNA, but this is DNA, so it will be AATG and
G, right? AUG will be ATG.
ATG, yes, because it will be the starting codon, yes, on DNA. It is exact copy. So actually, a messenger RNA is the exact copy of coding strand. So this is messenger RNA, and this will be the coding strand. So T actually, but it is U because this is messenger RNA, and G will be like this. Yes. So here, this is start point will be ATG on DNA. So first codon which will be made, it will be AUG. Then next this, it will keep on going. You got my point?
Okay. Yeah. Yes. Sorry, I'm just typing everything so to like. Yeah. So
no problem.
It starts with ATG or ATG, which gets
The start point is ATG.
ATG. Okay.
ATG is present with the coding strand, you remember?
Yes, it's present with the coding strand. So in the opposite strand will be
opposite strand of DNA will be containing what?
TAC.
TAC or TAC. Even you can remember this TAC. You can say the start point will be TAC, because we are talking in terms of
template.
Template strand.
Yeah. Talk in terms of template.
So it start, so at start point on template strand, it will be tag.
Yes.
Isn't it?
Yeah.
TAC.
Okay.
Okay. So first is initiation. Initiation involved promoter, RNA polymerase, and a start point. They combine together. Correct?
Yes.
And one strand act as a coding, the other strand act as a template strand. Then the two strands they get unwound. Yes, the breakup because of helicase. They have not mentioned helicase here. Yes, so
Yeah,
it is high level. So no need to mention this thing. DNA helicase is responsible to break down the hydrogen bond between two DNA strands, isn't it? So they get separated from each other, and this is known as transcriptional bubble.
Transcriptional bubble.
So this transcriptional bubble is moving next until the process of transcription is completed. So once the process of transcription completes, this is known as termination. So actually transcription start with initiation, then elongation, yes, and
yes,
then last one is the termination. Termination happens when the termination will happen. This is detail is there the next slides. Okay, when the termination will happen, when stop codon will come, isn't it?
Okay. Yeah, termination.
The termination will come once the stop codon will come, isn't it?
Yes.
So if the stop codon comes, the termination will start to take place.
Okay.
Now I'm moving to the next slide. So now this is the next slide is molecular components of transcription. So molecular components, this is RNA polymerase, RNA, correct? Then which pries the DNA strands apart and joins together RNA nucleotide, yes? RNA complementary to DNA template strand, yes?
Yes.
So messenger RNA is complementary to template strand. Please remember, messenger RNA, the RNA form, this is complementary to
template strand.
Template strand, and it is exactly the copy of coding strand. Okay. We call it mirror image, exact copy of. So messenger RNA is actually the exact copy, mirror image of coding strand. But messenger RNA is that the complementary strand for template strand.
Template. Okay. Messenger RNA is complementary for template, but
yes,
is the coding strand.
Yeah, a copy of a coding strand, mirror image of a coding strand. And then obviously for RNA, it will be T will be replaced by uracil. So this is very easy. So I'm not going into that detail. You know it, yes? Moving to the next. Now, the same thing, initiation of transcription at eukaryotic promoter. What is promoter region? Promoter region is a region where the initiation of transcription starts. Promoter is a protein which actually guides RNA polymerase to attach here and start the process of transcription, isn't it?
Yes.
Yeah, promoter, look here, exactly they have said, promoter signal the transcription, correct? Mm-hmm. Start point. So this is the start point. For what? For
RNA
polymerase. So it signals RNA polymerase. This is the start point. Come join here and start the process of transcription.
Okay.
Now there are some proteins. Look here. Transcription factors mediate the bonding of RNA polymerase and the initiation of transcription. So transcription factors, they help attachment of RNA polymerase to the template strand and they start the process of transcription. You understand?
Yes.
So what are transcription factors? They are assistant proteins which help to whom?
Assistant protein.
RNA polymerase to attach here and start the process.
Moving the RNA polymerase. Okay. Yes.
Correct. So the completed assembly of the transcription factor and RNA polymerase to bound to the promoter called transcription initiation complex. So during transcription initiation complex, what are the things involved? Can you tell me? One is promoter. Second is
one is promoter, other one is
promoter,
transcription
one is
factor.
transcription factor, and third one is RNA polymerase.
RNA polymerase.
So once they combine together, so they actually, you know what? They have specified the promoter is known as Tata box, TATA. Like, you know, TAC was what? What was TAC? TAC was the start codon.
Yes, start
TAC
codon, yes.
On template strand, correct?
Yes.
So this promoter, it is actually, we call it Tata box, TATA. There are 4 nitrogenous bases, yes?
Yes.
So this is actually very important. So before the starting point, they call it upstream, and this is downstream. Downstream, what is made? Messenger RNA. And upstream is what? The promoter.
Promoter.
And promoter is distinguished by a Tata box, TATA box.
Okay.
That's all.
So the promoter will have a TATA box.
Yes, TATA box. Okay. It is their distinguishing feature, Tata box.
Okay. Yes.
Okay.
Yeah.
Now next is this part. Already you know the same story that is going on. The same thing, like what is happening. These are 2 template and coding strand. So this is messenger RNA. You see, if you look at here, the messenger RNA is the exact copy of coding strand. AATT, yes, GG, 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 O. Obviously, T will be replaced by, it should be considered as T, yes, U. So exact copy, correct? And then these nitrogenous base, then, so this is transcription, and then this translate into protein. That's all. Here they are using, this is interesting. For some books, they use coding strand or non-template strand or sense strand.
Okay.
And some for the template strand, there are 3 names: non-coding strand, because it does not code for anything, or template strand, or anti-sense strand.
Okay.
This already I taught you in O levels.
Yes.
This was additional thing actually, you know what I taught at that time. Okay.
Yeah.
Now next thing we will be talking about this now. You are done with this, yes?
Yes.
So I'm removing all. I'm moving to the next then. Okay, next is this slide. Now, the same thing which is happening, you just go through with this reading as RNA polymerase move along the DNA, it unwind or untwists the double helix. Actually, it is not untwisting. For untwisting, what is used? Helicase.
Yeah, DNA helicase is used for untwisting.
Yes, DNA helicase is responsible for uncoiling or untwisting. But anyway, we have to go with this because they are using a generalized term. So this is happening. What is happening here? This is actually elongation process. So it keep on untwisting, keep on reading, and it is moving from 5 to 3 direction. So the transcription progresses at a rate of 50 nucleotides per second in eukaryotes. This is very interesting. So this is very, very high speed process. 40 nucleotides are joined together in 1 second. A gene can be transcribed. This is also very interesting. A gene can be transcribed simultaneously by several RNA polymerase. Now look, one RNA polymerase attached here, yes?
Yes.
It will move. Once it is moving next, behind here, again one RNA polymerase will attach. So simultaneously, it will make one messenger RNA. This is coming, it will again, because it is untwisted, it will get twisted again. Again, this will come, it will make the untwist. You understand?
Yes.
So they are, so simultaneously, it might be happening. This is very interesting, very important point. What is the advantage of this thing? The messenger RNA will be made at a very, very high speed. So maybe same messenger RNA, same type of messenger RNA you will make within 1 second. Otherwise, what will happen if this RNA polymerase is reading like this? Yes, finished. Then again it has to come back. Again it will restart. Again it has to come back. Again it will restart, isn't it?
Yes.
It is not happening like this. This RNA polymerase, it will keep on moving forward. New RNA polymerase, it will attach again from the start point. Again it will. So in this way, this is a kind of a sequence, one after the other, a kind of chain. Easy, yes?
Okay, it still takes some time if I keep, practicing.
No problem, but you are getting it, yes?
Yes, I'm understanding better actually.
Okay.
Yeah, okay. Yeah, we can go to the next slide.
I'm going to the next now. Termination. Once the termination, I told you what is the basic signal for the termination?
The stop codon.
Stop codon. So initiation is always there. For initiation, there are 2 or 3 things. One, start codon is there, isn't it? And the promoter region is there, yes?
Promoter region, start codon, yes.
Promoter or promoter region.
Promoter region. Yes, promoter region and start codon.
Yeah, promoter, or we can call it promoter region. We can call it promoter. Okay.
Okay.
And so in the beginning, this is their signal. Yes, these are the signals. Promoter actually signals transfer RNA. This is the start codon. So start from here. This is the starting point. Yes, it helps transfer RNA to attach to the start codon, isn't it?
Yes.
So similarly, the stop codon is actually giving the signal to RNA polymerase. Your work is done. Please leave. Isn't it? Because stop codon is not coding for any amino acid. So in this way, there will be the complex of DNA template strand and coding strand and messenger RNA. They will get separated from each other.
Okay. Yes.
This you understood, yes?
Yes, I understood.
Okay, now mechanism of termination are different in bacteria and eukaryote. This is interesting. For your level, this you require. Okay. Now how the this termination is different in bacteria. In bacteria, look here, this is the messenger RNA which is made. Okay, it is increasing, isn't it?
Yes.
This messenger RNA backside, it is already made. Yes.
Yes.
So once it is made, immediately the transfer RNA will start reading it and start making the protein.
All right.
It is very interesting. So because in eukaryote, there is no nucleus, correct? The messenger RNA is made. So first the transcription is taking place. Simultaneously, translation will also start to take place. Okay. So this is transcription is going on this way. And behind this translation also started.
Is following. Okay.
Yes, but in the eukaryotic cells, what will happen? Because transcription takes place inside the nucleus. So messenger RNA, which was made inside the nucleus, first it has to go out to the ribosomes inside the cytoplasm.
Okay, messenger RNA has
transcription. Okay. Messenger RNA is formed inside the nucleus. So this messenger RNA in eukaryotic cells, it has to leave through the nuclear pore and then it will reach to the ribosomes, isn't it? So the protein synthesis is clearly divided into 2 phases. One is the nuclear phase, which is transcription, and the other one is translation, which is the cytoplasmic phase.
Okay.
Okay, and one more thing, in bacteria, polymerase stops transcription at the end of the terminator, correct? Okay. And the messenger RNA can be translated without further modification. Immediately, the messenger RNA will be translated. Here in eukaryotic cell, for example, this messenger RNA is taken, has been made. Then there is one DNA splicing is there or RNA splicing is there. You know this? in A level.
Yeah, I think we did this in
Spliceosomes or spliceosomes, isn't it?
Yes.
So what this is messenger RNA. This messenger RNA, some of the RNA, they don't code. Some part does not code for anything, like introns.
Okay.
They are non-coding nitrogenous. They are non-coding parts. Yes. So they will be removed. They will be cut down, and only exons they will be joined together, isn't it?
Yes, neutrons and exons.
So what actually, even the messenger RNA which is made in the eukaryotes, still it is far from translation. First, it has to cut down. It has to get removed from the introns. So introns will be removed out of this messenger RNA. Only then this messenger RNA will become active for translation.
Okay. So it has to be removed. Yeah, it has to be removed for it to become active.
Yes, so because it is to become active, this is just like you can say like this is actually we have to remove it because then it is actually completely transformed into exact protein what we require.
Okay.
Okay. So intron are what? These are the parts of the sections of DNA or RNA which does not code for any amino acid, any protein. And exons, exons they are expressing due to they express for one particular protein, isn't it?
Okay. Yes. Sorry.
Yeah. Now you understood all this.
Yeah.
So eukaryote RNA polymerase 2. So there are there is DNA polymerase 1, DNA polymerase 2, yes. DNA polymerase 3. So similarly, you just have to remember RNA polymerase 2 is there. Okay, transcribes polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotide past this this we will be discussing next.
Okay.
Okay, polyadenylation. What is polyadenylation? Adenylation, it means adding more AA, adenine nucleotides.
Oh, adenine nucleotides. Okay. Okay.
Yes. So actually, adenylation mean polyadenylation mean this messenger RNA is made. Yes. After that, what you do, you add some this part you add. This is this is tail. So it is all is made up of A.
Yes.
Poly A tail. We call it poly A tail. So processing is longer in eukaryotic cell. So directly you cannot copy for translation. Okay. So number of checks and balances are there. You understand?
Yes.
And polyadenylation, this AA poly A can be from 10 to 35. Long tail, poly A tail. Okay?
Yes.
Okay, I'm removing this and moving.
Yeah, we can go to the next slide. This isn't difficult. At least this slide.
This one?
Yeah, this is fine. This one is like how bacteria.
You understood this part, yes?
Yeah.
Elongation of proceed and until RNA polymerase copies. So you can, this is actually this one. This one you just, the GC region in the RNA forms a hairpin loop pulling the RNA molecule away from the DNA.
Okay.
Okay. Then the weaker bond between the U's and A's of the template strand breaks, releasing the RNA.
Okay.
The termination signal, look here, elongation of the RNA chain proceed until RNA polymerase copies a sequence called termination signal. The GC region in the RNA forms a hairpin loop. So this is a loop is formed. Once this hairpin loop is formed, what does it mean? That it is actually, this is the termination.
Okay.
Okay. Now, the point what we have to remember, like, termination of transcription in bacteria. This is bacteria. Bacteria have something little bit different. Like in eukaryotes, this is poly A, yes?
Sorry. Yes, poly A.
In eukaryotes, this is poly A tail, yes?
Poly A tails, okay.
In bacteria, elongation of RNA chain proceeds until RNA polymerase copies a sequence called termination signal. What is a termination signal? The GC region in the RNA, wherever the GC will be there, it will make a hairpin like this.
Okay.
Can you see my cursor?
Yes.
Okay. So it will make a hairpin loop here. So it will pull the RNA. For example, if you are making a loop, so it will pulling the RNA molecule away from the DNA. So it will, because it was a weaker bond, so it will be pulled out from here. If you understood, this is not that important, but if you understood, I will move to the next slide. Little bit, you got it?
Yeah, I understood a bit.
Bit or completely. Look in the bacteria, what happens wherever the GC will be there, wherever the GC near to this
anywhere there is the GC.
Yeah,
it
will make a loop. Look at the loop now. It will make a loop. It will start to unwind. It will start to wind up like this. Like you can see the bulb shape.
Yes.
A bulb shape. So it will start to wound. So once it will start to wound up, okay? It is winding up, yes?
Yes.
So once it is winding up, it will pull from here. From here, it will pull. So this messenger RNA will detach from the DNA.
Okay.
Template strand. So it gets separated. So look here, it gets separated. So they are showing it here, this part. So you just remember GC hairpin, a loop is made and that loop pulls the messenger RNA out of template strand complex. So messenger RNA is separated from the main strands of DNA.
Okay, mRNA separated from main strands. Okay.
Okay, now if you see termination of transcription in eukaryotes, in eukaryote this is different. What happens? They are using energy, ATP they are using. Look. So stop codon will come, correct?
Yes.
So now for polymerase 2, the transcripts are cleaved and broken down at specific site before transcription ceases.
Okay.
The cleavage site is 1035 nucleotides downstream of a poly A tail, AAU sequence in the RNA. Okay. The cleavage site of polymerase 2 transcript is also the site for addition of poly A tail.
Okay.
Okay. Yes. And this string of 250 to 250 adenine nucleotides. We learned it already. This poly A tail can be made up of 50 to 250 adenine nucleotides. Added on the 3 direction. This is 3rd and this is 5. So I told you always addition will take place on number 3 position. Yes.
Yes.
So they make poly A tail, and this poly A tail is made with the help of poly A polymerase. This addition of tail is done by one more enzyme, which is poly A polymerase. Now RNA polymerase is gone. Its duty was up to here, this brown color. Once the yellow color is starting and it is adding one tail, so what will happen? Another enzyme will come, which is poly A polymerase. So it will add 50 to 250 adenine nucleotides to the end of this after the stop codon.
Okay.
That's all. There they are pulled, isn't it? In eukaryotic cell because of hairpin.
Yeah.
Sorry, in bacteria.
Yes. So hairpin.
So it will, in the prokaryotic cell, there will be hairpin will be made and it will be pulled. Here what is happening? Here the ATP energy will be used and then it will be added with the poly A tail. Okay, and then it will get separated. That's all.
Okay.
Here, one more interesting thing is there. cleavage 10 to 15, 10 to in fact 35 nucleotide. Now, wherever, this is the stop codon here, yes?
Yes, stop codon.
And there will be AAU, this one, this sequence will always be there. After this sequence, there is again, you have to keep they keep a distance of 10 to 35 nucleotides. Okay. And after that, they add a polynucleotide, poly A tail. Once we will move up next, then maybe you will get more in detail. Okay. So this part is not actually coding for any protein. So this will be intron. Whatever is added, it will be intron. Later on, it will be removed because it is not coding for any protein. So that is why you just remember this part.
Because it's not coding for any.
Next slide.
Yeah.
Now, this already we learned. In prokaryote, translation of messenger RNA can begin before the transcription has finished. So once the transcription has started, immediately translation also starts. Very interesting statement here. In prokaryote, bacteria, the translation of messenger RNA can start before the transcription is finished. I already told you.
Yes.
But in eukaryotes, obviously, because it has clearly divided into two parts, so transcription is taking place inside the nucleus. So first the transcription will take place, that messenger RNA may come into the cytoplasm, only then the translation will start.
Okay.
Moving to the next. Now we have to go for, there are some enzymes which are required for RNA processing.
Okay.
So what is pre-messenger RNA? Pre-messenger RNA is an RNA which contains both exons and introns.
Introns.
Immediate introns and exons, they are together.
Yes.
So you have to cut introns and you have to draw, you have to unite the exons together. So then it will act as active messenger RNA.
Okay, active
so
messenger
look
RNA.
Here, we are cutting different sections. Certain sections are cut out and then so remaining parts are sliced together. So after, first is pre-messenger RNA. Then pre-messenger RNA, it will be spliced into different parts. So during splicing, what is happening? Intron parts are removed.
Okay.
Then exon parts are united. This is next topic. We will be doing it now.
Okay.
You understood? Yes?
Yes.
Shall I move next?
Yes, yes, sir.
Shall we take a break of five minutes?
Yeah, yeah, I'm fine with taking a break. So
we will take a break of just 5 minutes, okay? And then I will be joining you back. You can pause the recording, okay? Okay, you started the record, yes?
Yes.
Okay. Now the position is alteration of messenger RNA ends. Now the end should be altered both from the top and from the bottom, mean from the cap, from the head and the tail. Okay. The tail is poly A tail, yes? We at 3 end, there is at 3 end, there is a poly tail, yes?
Yes.
What do you think? What will be the purpose of this addition of a tail or a cap?
It's to make sure like nothing is missed in a way.
No. It is actually, for example, if this is the messenger RNA, okay, and this is 5 side, this is the cap, and this is 3 side, which is the tail, poly A tail is added. So actually, this is for protection. Because once this will be moving from the cytoplasm, yes, so there are different kind of chemicals. So they will not directly come in contact with the head or the tail. They cannot alter them. So they are actually, guards. They are the guards for the messenger RNA. So it should be, transferred to the ribosomes safely. You understand? We are capping it. So this is for the safety. First, why we are doing it? They seem to facilitate the export of messenger RNA to cytoplasm. Yes, facilitate first to protect, and last one to help ribosomes attach to the 5' end.
Ribosomes attached to the 5' end. Okay. Yes.
Okay. So now actually what is happening? This is a start codon. Yes. This was what? This was the promoter region, and this is 5. This is a cap, which is actually GTP. Like ATP is there. Yes. Fatim, you understood? Yes. So this is GTP.
Yeah, GPP. Okay.
Guanine triphosphate. Okay. So, and here is poly A tail. So this already you know what is the purpose.
Yes.
Okay. I'm moving to the next. Okay. The next slide is. If please tell me if you have somewhere, if you have any kind of doubt. Why I'm presuming everything is getting clear, so we are moving faster. Yes. So we cover it as much as possible. Splitting genes. Already you know this thing. RNA splicing, isn't it?
Yes.
You know this concept.
Yes, introns and
exons. There are introns which don't code for, this is non-coding region, correct?
Yes.
This is the non-coding region. It does not code for anything. So what we need to do, we have to, the exon, this is non-coding region, for we have to remove it. And then exons, they are coding region. They are the region which actually expressed, which is eventually expressed, genetically expressed. So once you remove introns, and then introns we will be removing, okay? Once you remove the introns, and then this part, this is exon, and this is also exon. So you will be joining them together. So it will be taking one active segment of messenger RNA, which is only containing exons, and it will be coding for one specific protein. It will express for a protein. Any problem?
No problem, sir.
Okay, I'm moving next. Okay.
Okay.
So I'm moving to the next slide then. Now, next slide is this one. This is a little bit, challenging. There is a one term that is known as spliceosome.
Spliceosome. Okay.
Spliceosome or spliceosome is a better kind of word. Yes.
Yes.
So once we are talking about spliceosome, so what is spliceosome? Spliceosome is actually snRNA. snRNA stands for small nuclear RNA.
Okay.
And then what happens? This is one protein is there. This protein is small nuclear RNA protein, snRNP.
Okay. SNRNP.
Now, SN means small nuclear ribosomal ribonucleic protein. RNA, ribonucleic acid, yes. So this is ribonucleic protein.
Yes.
Correct?
Yes.
If you see it here, what is the function of the spliceosome? Actually, their function is to cut the introns. Introns, they move out here. And so they convert the pre-messenger RNA into one active form of RNA, which is containing only exons and exons joined together. So that is why we call them spliceosomes. They splice the RNA. They cut it into small pieces and then they join them together. So there is one more thing. This snRNAs are actually ribozymes. What is the meaning of ribozyme? Ribozyme means an enzyme which is responsible to break down bonds.
Yes, ribozyme. Okay.
They break bonds.
Okay, yeah. So
either they will
that's how
break down or making both.
So So that's how
joining
they
or
remove introns and they add the exons, right? They
yes,
bring the
yes, yes.
exons
So
to the
that is why we
junction.
are calling it spliceosomes.
Okay.
So what is a spliceosome? Spliceosome is the combination of RNA and protein. Which
Okay.
RNA? snRNA. And what is the protein? It can be snRNP. Combination of this and this. They combine together, they make a spliceosome. One big body is made. This some means body, okay?
Yes.
Soma, nucleosomes, or lysosome means somes mean body, correct? So they make a body. This body is responsible to cut this part. So this body is containing what? RNA, which is snRNA, and what is the protein? snRNP.
snRNP is the protein and sRNA.
Yes,
Okay.
small nuclear ribonucleic protein, snRNP. You just
Okay.
remember the abbreviation name, so you will remember
Yes.
this part. And snRNP is responsible to make one enzyme. What is that? They act as, they mean snRNA act as ribozymes. What are
Okay.
ribozymes? They are responsible to break down or make or
Okay.
synthesize, catalyze some bonds. Any problem? This was one of
No, no
the, this was
problem.
the little bit tough
Yeah.
topic, yes? So I
Yes.
hope, Alhamdulillah, you understood this part, yes?
Yes.
Yeah, okay, I'm moving next now.
Okay.
Okay, now this is general, ribozyme. So already you know what is the function of the ribozyme, so I'm not going to go for the detail. You can read by yourself. So ribosomes are catalyst RNA molecules that function as enzymes. Three properties: it can form three-dimensional structure. Some bases in RNA contain functional groups that may participate in catalysis, and RNA may hydrogen bond, okay,
Okay.
with other nucleic acid molecules.
All right.
Okay. Now, next thing is, this is again, this is one of, again, a different thing and difficult thing. Now, look, the function and evolutionary importance of introns.
Okay.
Now, what is happening now? So, for example, we have in this one, skipped exon is there, yes?
Yes.
From here to here, exon is skipped. So it can make like this. This is
Okay.
interesting. Okay, please make note of this thing. This is very important.
Okay.
I will try this. I give you in my this one. Look here. There are two possibilities. Either both are, three of them, all of them, they are skipped. So it will take this shape. Yes, exon and exon.
Okay.
Then there is a second condition in which this major exon, intron and intron is removed, but small portion still it remains here. So now you
Okay.
see there is a difference in length, yes?
Yes.
So once there is a difference in length, so it has a kind of evolutionary importance. What are introns? Introns, they don't code for any particular kind of protein, but
Okay.
their exact function we don't know, yes? What
Yes.
exactly is their function?
That's not
What
unknown.
factor they control? So that is why some kind of changes may take place, and you understand? So in this
Yes.
way, from one pre-messenger RNA, they can generate two different lengths of messenger RNA, which is active. You got
Okay.
it?
Yes.
So in this way, what we are saying, the functional and the evolutionary importance of introns. So introns are really very important in the process of evolution. So how, like introns, what kind of function they are controlling, how one person is different from the other. So these, they will be giving that kind of changes. Okay. So
Okay.
I will just read through.
All right.
Uh, exons typically encode proteins, which is actual domain. We know it.
Okay.
Like they will be coding one particular protein. We know it. So that they have one particular domain. They cannot go out of this. So these domains, so in this case, there are how many domains? There are 2 domains, 1 and 2. We call them isoforms. Iso mean they are similar. Similar in what? They are similar in exons, but
Similar
yet
in
they are
exons,
different
yes.
from each other.
Different from each other, but similar in exons. Okay. Okay.
Yes?
Yes.
So now here we have to look here. A consequence of alternative splicing. Alternative splicing. You know what mean? What exactly? Here is different. Here is different. Here is different. This is skipped exons. Yes.
Okay. Yes.
Alternate spliceosite. Yes. 5. Then alternate 3 splice site.
Okay.
Then what they are giving different kind of retained introns.
Oh.
So intron is retained.
Okay. Intron
So
is
just
retained.
go through with this and mutually exclusive exons. So here, you know what, introns are removed and they are different. So if so, what is their importance? This is pre-messenger RNA and this is the active messenger RNA. Yes, all of them. They are. So what is the importance here? So here introns, they play a role in evolution. Sometimes they become part and they actually, but mainly each exon is having a particular domain that you just remember.
Okay.
Achha, what is the meaning of this thing? The proteoma or proteome is larger than the genome. What does it mean? Proteome is larger than the genome. What does it mean? According to the gene, the size should be like this, yes?
Yes.
But the protein size is like this. How? You understand? Originally, it should be like this. It should be this one. But proteome means a protein which is made, it is larger than it should be. Protein size is larger than it should be. So what will happen if the protein size is larger than the normal? So some kind of changes, mutation or evolution has taken place. Some people, they will be having, for example, I'm just giving you a literally layman explanation. Look, some people, they will be having more, flesh on their biceps. Why? Because the bicep muscles, they contain larger proteome.
Okay.
This you understand. What is proteome? A protein body. This messenger RNA should make this protein. For example, this messenger RNA should be making this size of the protein, this kind of protein, like this. Okay? But instead of making this kind of protein, it is making a kind of protein like this. So larger than the normal. This happens because of this splicing introns and all. Different sizes.
Okay.
That's all. So this is evolutionary importance of splicing. Once I will be going next, then you will understand this also. Okay? I'm leaving it as such. If some misconceptions are there, so once you will be going through with this again, you will get it clarity. You will get the clarity. Okay? I'm moving just for the next slide. Okay? So here, if we move for the next slide, this one. They remove the exon. Exon is here. Yes, green is there, or this one, orange is there, and purple is there. Yes. So these 3 proteins are the exons are there. These are 3 domains, correct?
Okay. Yes.
These 3 domains, they can join together differently, so they will give a different kind of protein.
Okay.
If protein instead of joined with orange, it joins with blue, and then orange is joined with this end of blue. So it will be different. You understood?
Yes.
So this is actually, so different exons code for different domains in a protein. Yes, exon shuffling may result in evolution. This is the most important stuff.
So the shuffling of exons will result
Yes.
in evolution.
Yeah, that is what the thing that we learned in the last lesson, last slide. Okay. If we
Okay.
shuffle them, so they will be, actually exons are the same. One exon, exon 1 will be making this domain, 2 will be making this domain or this protein, and 3 will be making this particular protein. But assembly of the protein, if it changes, if we have bricks, yes?
Yes.
So you can make different designs. If you have a red brick, you have green brick, and you have purple brick. If you can, this is your decision how you arrange them. So if you change the arrangement, their shape will change. Protein, they are very specific, yes? So that is why this character is having shuffling of exon result in evolution.
Okay.
Moving next now. Okay, now next is, you know already the transfer RNA, yes? Transfer RNA, they are highly specific, correct? Now, transfer RNA, why we are calling them the transfer RNA? Because they are specific for one particular amino acid. Now, the thing that you have to remember, a transfer RNA has what parts are important with the transfer RNA? Transfer RNA has one part which contains what? Amino acid. Amino acid is always attached to number 3 side. Please, this is the most important thing. Amino acids are always attached to 3 side of transfer RNA.
Okay. Amino acids.
Each transfer RNA is made up of almost 90 nucleotides. Important thing.
90 nucleotides. Okay.
A transfer RNA molecule consists of a single RNA strand that is only about 80 nucleotide long.
Okay.
Correct?
Yes.
So amino acid, obviously, what is the function of the transfer RNA? They transfer the amino acid. Amino acid will be attached where? At number 3 or hydroxyl part of the ribosome or nucleotide. I showed you the diagram. Yes, I sketched that diagram. Number 3 was OH group. Yes.
Yes.
So amino acid will be attached there. So what will be at number 5? What will be here?
Number 5 will be the phosphates, right?
No, yes, sorry. It will be phosphate, Shabash. It will be phosphate. Good. And one more thing is there. Now, here, a transfer RNA has one part which has complementary nitrogenous base for messenger RNA that is known as anticodon.
Okay.
Correct? You know this concept already.
Yes.
Correct? So anticodon. So this anticodon, it will stick to the transfer RNA. So if it is complementary only, then it will stick to the transfer RNA, and then it will be holding its amino acid. That amino acid will be used to make a polypeptide. We will be talking next.
Okay.
Okay, I'm moving to the next. Is there, and you just remember this. This is clover leaf shape is there. Yes,
Yes,
transfer
clover.
RNA has a clover leaf shape.
Yes.
Okay, so on one side there is one anticodon. Opposite to that anticodon, on the number 3, it has amino acid. This is important.
All right.
Okay, I'm moving to the next. Now, already this is a new thing. Now, there is one there is one enzyme which is responsible. There is one enzyme which is responsible to attach amino acids to the transfer RNA.
Okay.
This enzyme is called amino acid transfer RNA synthetase.
Amino acid. Okay.
So amino acid transfer RNA synthetase
Okay.
So this is responsible to make sure that one particular amino acid attaches itself to a particular transfer RNA.
Okay.
I told you there are total how many essential amino acids? If by any chance you can remember, there are
20,
almost 20 essential
20 essential.
amino acids. Yes, there are 20 essential amino acids. So I told you each amino acid is specific for one specific transfer RNA, correct?
Yes.
So obviously, once there is one for each transfer RNA, there is a specific amino acid, correct? Similarly, for each amino acid and transfer RNA, there should be one specific amino acyl transfer RNA synthetase also.
Okay.
Please, this is additional thing that you are remembering now. This you didn't discuss this thing in grade, in your school, college. Okay, so now till now you are knowing that a transfer RNA is specific for specific amino acid. So if there are 20 essential amino acids, there will be 20 essential transfer RNA. So there will be 20 different type of transfer RNA. But now we learn for each transfer RNA, since each transfer RNA is specific for one particular amino acid, so this attachment will be monitored by one particular enzyme. So for every transfer RNA and amino acid to combine, there is a helper. Which is that helper? Amino acyl transfer RNA synthetase. So for each reaction, there is a specific enzyme. So there will be 20 different enzymes. Amino acyl transfer RNA synthetase. Function, you know it now.
Yes.
Okay. Now this additional information you can read, this wobble and all you can read. We learned we have done the basics. Okay.
Okay. Yeah. This one.
This is nothing. This is nothing actually. Okay. We are moving to the next slide now.
Okay.
The next slide is now if you see here, now they are showing how this transfer RNA synthetase is helping us to, it is helping the this transfer RNA to attach to one amino acid, correct? First of all, we have to remember this attachment of amino acid with transfer RNA, it need energy. So there is one ATP is used here for this association. So this whole part is actually transfer amino acyl transfer RNA synthetase. This is enzyme. Correct?
Yes.
This enzyme, what it will do? It will be attaching the this transfer RNA with one particular amino acid. So this is amino acid and this is transfer RNA. So it will get attached with the help of ATP. Then they are using different kind of terms. You know, ATP is actually in this process, ATP is converting into AMP. ATP has changed into AMP. Just a minute, okay? It directly changes into AMP, adenosine monophosphate. Mean loss of two phosphates take place. So once the loss of two phosphate take place, so more energy is released.
Okay.
Remember, highest energy is present with ATP. Look, this is interesting. This because I was teaching one student in AP biology. Okay.
Okay.
So that kid was having this thing. So I will just look. For example, if we have this, just 1 minute. If we have this ATP, okay, I'm making ATP. Correct? And then it will be having, I just do it a little bit faster. So there will be one phosphate, second phosphate, third phosphate, isn't it? I'm just giving you example. I just wind it up fast, okay? So if this is 3 phosphates are there, so this is one bond, yes? This second bond, and this is the third bond, yes?
Yes.
This bond will never break. It is unbreakable. So if you break this bond, highest amount of energy will be released. If you break this bond, again energy will be released, but it will be less than the first bond which were broken. So these two bonds are actually energy bonds. But the last phosphate cannot be removed from the nucleotide A and P. So these two bonds are broken down, so more energy is released. This energy is used by amino acid transfer RNA synthetase to make this complex, to join this transfer RNA and amino acid together. That's all.
Okay.
Here they are using this term. This is weird kind of term. Pyrophosphate. Yes.
Okay. Yes.
Pyrophosphate. Once double phosphate are removed, they are double together. Together two phosphate, they are pyrophosphate, and then they are broken down into single phosphates.
Okay.
Okay. The specificity of genetic code is mediated by codon-anticodon interaction. You know it, yes? The transfer RNA that binds to messenger RNA code specifying a particular amino acid must carry only the amino acid. Mean it has to have a one particular amino acid. That's all. So this already I told you in the previous slide. Okay.
Okay.
So every amino acid is specific one particular
protein.
Amino acid specific one particular transfer
codon.
RNA.
Codon. Yes.
Transfer RNA.
Transfer RNA.
Yes. Even codon, this is in general you are talking, yes?
Yes.
In general, but since we are talking about transfer RNA, so one particular amino acid is specific for one particular transfer RNA, correct? Now, if you remember this, ribosome structure, structurally a ribosome is 50% protein and 50% nucleic acid.
Okay.
Which nucleic acid ribosome is made up of? This is known as rRNA. Like there is messenger RNA. Ribosomal RNA, correct? You remember this part? Yes.
Yes.
So that 50% which is there, that is ribosomal RNA, which is actually structural part. Bacterial and eukaryotic ribosomes have similar but significant difference. One difference that I told, if you remember, the size of ribosomes of bacteria, this is 70S, and size of eukaryotic ribosome is 80S. So eukaryotic ribosomes are bigger than prokaryotic ribosome. This is first difference. Okay, and second difference, these bacterial ribosomes, sometimes they are more vulnerable for one particular antibiotic. So for example, once we are taking antibiotic, yes, so it will kill the ribosome, but only the ribosomes of bacteria, but same antibiotic will not affect any of the eukaryotic ribosomes. So this is very important thing, this point.
Okay.
Got it?
Yes.
Okay. Now, shall I move next?
Yes.
Okay. Yeah, now next is, oh, sorry, this one. Now, again, I told a ribosome is actually made up of two subunits. You remember? If you can recall, we used to do it. A ribosome has two parts, larger subunit and a smaller subunit, correct?
Yes, up and lower.
Yes, larger subunit and smaller subunit, yes? So larger and the smaller subunit, yes? Now, in larger, you just remember, and these two subunits are joined together by magnesium ions. Please write down this thing. It is not there in your book.
Magnesium ions.
Okay. They are held together by magnesium ions. Okay. Now, one. Next thing, even it is not here also. Okay, second thing that we are doing now, this smaller subunit which is there, yes, it acts as a template for messenger RNA. Messenger RNA will get here like this. It will get attached to this smaller subunit.
Okay.
And once it will get attached to the, you can say the messenger RNA lays down on top of small subunit. And then this larger subunit, it comes on top of it and close it.
Okay.
Like a box. You put something into the subunit, you put one layer, and on top you put the second part. So, but in the larger subunit, this is additional thing. In the larger subunit, there are three components. One is A site, then second is E site, and then third one is A site, E site, and P site.
Okay.
P site, and then one more is there. What is that? A site. This is important. So it has three parts. Larger subunit have these three. Okay. A for additional site, P for protein, and E for exit. So larger subunit have three parts. One is, or three sites. One is E site for exit.
Yes.
Elimination.
Elimination. P site for protein.
E for protein, and A for additional. Mean this you just remember, okay? P site hold. This you already mentioned. So I'm moving next then. Okay. Shall I?
Yes, sir.
Are you okay? You are not tired?
I think like I'm pausing the recording.